Good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Accelerate Cell Culture Media Development with Automated Cell Analysis. My name is Sonia Hunt, and it's my pleasure to be your X-Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the Comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Nova Biomedical, who developed the content for this presentation. Advanced Instruments and Nova Biomedical are now doing business under the same brand, bringing together decades of expertise in analytical instrumentation, R&D, and global customer support. BioProfile FAST CDV is a key component of Nova Biomedical's cell culture analysis workflow, delivering rapid, automated, viable cell density measurements to support media development and process optimization. Designed to improve throughput and data consistency, FAST CDV enables efficient decision-making across early-stage development and upstream workflows. Now it's my pleasure to welcome and introduce our speakers for today's event. And first, I'd like to introduce you to Elizabeth Abraham. She is a life sciences strategy and product innovation leader with over 20 years of experience advancing product development and commercialization in cell and gene therapy, biologics, and regenerative medicine. Elizabeth currently is the Senior Global Product Manager for the BioProfile Cell Culture Analytical Platform at Nova Biomedical, where she focuses on enabling next-generation bioprocessing through advanced analytics. Prior to joining Nova in 2026, she held multiple global senior leadership roles and has authored 23 scientific publications and is an inventor on 11 patents. Now it's my pleasure to welcome and introduce you to Dr. Sofia Pizoa. She is a senior scientist on the media development team for Lucius Labs. Prior to joining Lucius Labs, Sofia received her PhD in cell biology, stem cell, and developmental biology from the University of Colorado, Denver, and her BS in biology from Syracuse University. Sofia joined Upside Foods in 2023 on the media development team and is now working with Lucius Labs, part of the Upside Group, to develop novel and custom cell culture media formulations. Now, with the introductions out of the way, it is now my pleasure to pass the mic over to our first speaker, and that is Elizabeth. So Elizabeth, when you're ready, you may begin.
Hello everyone, thank you for joining us. Today, our webinar is titled Accelerate Cell Culture Media Development with Automated Cell Analysis. I'm Elizabeth Abraham, Senior Global Product Manager at Nova Biomedical for the BioProfile platform that includes both the Flex and FAST CDV products. Before we begin today's session, I want to remind you to please submit your questions throughout the presentation, and we will address them at the end during our Q&A session. Nova Biomedical is a global life sciences company dedicated to improving patient outcomes by increasing productivity and ensuring quality across laboratory environments spanning bioprocessing, clinical diagnostics, and food and beverage. We're recognized as a global leader in osmolality testing and whole blood biosensor development, and one among the world's fastest growing in vitro diagnostics companies. We're also experts in cell line development technology and liquid handling quality assurance, all backed by a proven track record in instrument development and customer support across more than 90 countries. With that context, let me take you through how we got here. Nova Biomedical and Advanced Instruments each bring decades of innovation to the table. Nova pioneered whole blood biosensors in 1979 and launched the first BioProfile Flex 2 cell culture analyzer in 2016. Advanced Instruments commercialized freezing point depression osmolality technology back in 1955 and later expanded into cell line development and liquid handling through the acquisitions of Solentum, SAL Scientific, and Artel. In 2025, these two organizations came together to form a unified life sciences company, combining more than 120 years of expertise in biosensors, osmolality, automation, and bioprocessing. That foundation is what powers the BioProfile portfolio and the reliable day-to-day cell culture monitoring we'll be discussing today. This slide gives you a bird's eye view of the markets we serve and the breadth of our technology portfolio. Across bioprocessing, we support customers throughout the entire workflow, from cell line development and clone selection with Solentum and NanoSelect, our latest addition, to osmolality testing with Osmotec, liquid handling verification with Artel, and routine cell culture analysis with BioProfile platform. On the clinical side, we cover critical care, CSF cell counting, osmolality, and point-of-care testing. In food and beverage, we support dairy quality and pasteurization workflows. Today, we're zeroing in on our BioProfile platform, specifically the FAST CDV. BioProfile FAST CDV is the fastest, lowest touch route to reliable cell density and viability data, with cell counting performed in under 70 seconds per sample using AI-enhanced tripannBlu counts, using minimal volumes to provide accurate results from 100 microliters with no manual prep steps. The onboard automated dilution handles densities up to 140 million cells per mil, so high-density cultures need no rework. It provides walk-away throughput with the 32-position carousel or optional 96-well plate, freeing up operator time. RFID cartridge packs self-recognize on install, keeping maintenance simple and traceable. It is GMP ready, 21 CFR Part 11 compliance plus OPC connectivity provide flexible data transfer options. Shown in the bottom panel is a schematic of the steps within a FAST CDV workflow, starting with sample loading, either on the carousel or 96-well plates. The next steps are automated and all happen within the unit, whereby the cells are mixed with trypan blue and plated on an imaging chamber. The output contains a series of images that provide various parameters such as total cell density, viable cell density, viability, and average cell diameters, providing faster answers, less hands-on work, audit-ready data. An activity turns a FAST measurement into a usable, trusted result. Let's compare and contrast the workflow of the FAST CDV versus traditional pathways. For the FAST CDV workflow, in the bottom left panel, you load the sample, use the touchscreen to start analysis, and walk away. The FAST CDV analyzer has inbuilt computers as well as OPC compliance servers. The OPC UA is an open, standardized, secure protocol so IT and automation teams integrate without custom middleware. The benefits of the OPC server is captured on the bottom right panel in that it connects to MES, DCS, and LIMS with results published in real time. No manual transcription means fewer errors and no lag between measuring and acting. Data lands audit ready and supports 21 CFR Part 11 data integrity expectations. Now contrast that with the manual workflow shown on the top left panel, where the sample needs to be diluted with diluent, mixed with viability dye, transported to an analyzer, and results transcribed, adding delay and using error-prone methods. Automation plus connectivity gives faster, cleaner, and a more defensible process data. And we get to see that in action in the next presentation by Sofia Pessoa. Thank you.
Hello, and thank you for the kind introduction. Thanks everyone for tuning in today. I'm Sofia Pessoa. I'm a senior scientist with Lucius Labs, and I'm excited to talk today about our media development team and how we utilize the BioProfile FAST CDV to automate our processes and speed up the time that it takes our team to develop new cell culture media products. So here's a review of the agenda for today. I'll start by giving a short introduction into Lucius Labs, as I am sure there's quite a few of you who might have probably not heard of us before. And then I'll move on to present our use cases for the FAST CDV for our team. Our first use case will focus on facilitating media development using semi-automated tools and how the FAST CDV is integrated into that. The second part will focus on accelerating the analysis of multiple cell types. And then the last use case, we'll talk about the accuracy of the FAST CDV over a wide range of cell densities and how that's important for our processes. We'll conclude with final remarks, and at the end, we'll leave time for questions and answers. So I'd like to start with an introduction to Lucius Labs, as I'm sure many of you have never heard of us before. So our story begins with cultivated meat and upside foods. So we essentially spun out of Upside Foods, and the reason for this is that while developing cell culture media for our cultivated meat products at Upside Foods, this really forced us to drive down the cost of our cell culture media and develop our own options in order to reduce our production costs. So around 2019 is when we developed our cell culture media team here or at Upside Foods, and this was with the sole intent of developing our own cell culture media options. Around 2020, we had established our internal media library, our own proprietary formulations, as well as media manufacturing and analytics at Upside. In 2025, we launched Lucius Labs with the intent of bringing our cell culture solutions to the world and providing academic labs and industry labs with cheaper and better cell culture options. And now in 2026, as part of the Upside Group, we are working together to launch cultivated meat and bring industry-enabling solutions to everyone. So all of our hard work has led to a robust media library of proprietary formulations that work across a wide variety of cell types. We are able to deliver optimized performance by developing media tailor-made for specific cell types and processes. Our media library works across a wide variety of cell types, whether it's for cell agriculture or other bioprocessing. Our team also includes in-house analytics with a dedicated analytics team to rapidly analyze media samples, and we work hand in hand with them, whether it's to analyze trace elements or spent media. We are also able to adapt to multiple modalities, with our team having extensive knowledge and experience adapting cells into animal component-free, serum-free, or suspension modalities. Our media has been capable of working with a wide variety of these processes. And lastly, we also have internal prototype manufacturing. So we're able to deliver custom and proprietary media prototyping and include dry powder milling and own the process from the raw material to the final product. So all of this has led us to be able to deliver industry-enabling solutions, whether it's for cell therapy and stem cell processing, or gene therapy and developing lentivirus or AAV with HEK, vaccine development with non-mammalian expression systems, and of course, cultivated meat. We have a robust media library and team that's dedicated to developing these products. So more importantly for the talk today, I'd like to focus on how we accelerate this media development and how we're able to move from a concept into a final product within 1 year. So the first part of this will focus on facilitating media development using semi-automated tools, and an important part of that is going to be the FAST CDV. Before I get into that, I'd like to start with media development and exactly what that is. So just a little bit more background information for anyone who's not familiar with media development. But essentially, our focus is to provide cells in vitro with a precise combination of nutrients that they require. So there's a lot of things that go into cell culture media. There's a lot of nutrients that cells require, and there's an exact combination of these nutrients that has to be experimentally determined. So in this graph, I just wanted to highlight the glutamine pathway and how many different steps are involved in this, and how many different amino acids are also involved with it. So whether they're being consumed or produced, at each stage, there's an exact ratio and combination that cells need in order to provide the cells with the sufficient amount of energy so that they can sustain their metabolism during growth and production in each of these phases. So our team is dedicated to figure out what these combinations are, when and how they need to be supplied in the bioprocess. And the way that we go about this is to take 3 steps, with the first starting with screening these components. So this can be many different components, of course, amino acids in this case of looking at glutamine synthesis. So we start with a screen, and then we go on to refining these components. So essentially removing anything that might not be essential, or focusing only on the things that are required, and then optimizing these. So optimizing the concentrations or even the form of which it's delivered can be critical into developing a really robust media. So what we utilize very heavily is design of experiment, or DOE. This allows us to quickly screen several variables at once. So instead of taking, for example, a path of one factor at a time, we use multifactorial optimization. So this allows us to screen many factors at once, where we start at the screening step and determine what the most important factors are. And then we move on to an integration state of refining. So focusing then only on the factors that are providing the most robust response, or eliminating any factors that are perhaps detrimental to the process. And then from these chosen factors, we go on to optimizing them. So looking at the concentrations or the factors and really optimizing this using response surface methodology to determine where that sweet spot lies in the dosage. And if some of these examples, of course, are amino acids, could also be fatty acids or iron sources. Growth factors is another one where these things usually take a lot of screens and refinement in order to determine what is the correct dosage, how should these things be provided to the cells. Of course, this can take a lot of time, so DOE is really important for us to do this very quickly, and we rely on this heavily in addition to instruments like the FAST CDV. So this is a typical experimental workflow for our team that enables us to develop media very quickly. We usually have a lot of variables, which creates a lot of conditions, so we need tools to move very quickly. Our typical workflow starts with design of experiment, so utilizing something like JUMP to develop a DOE, and then move from this design experiment into data collection. We rely heavily on instruments like the FAST CDV that do automated data processing so that we're able to rapidly move to data analysis and analyze the data the day that as it's coming in. With the FAST CDV, we are able to automate sampling and enable rapid data analysis. The FAST CDV works with either single sample cups or 96-well plates and quickly switches between the two without the need for removing machine components. As shown in this video, there is a quick option for either 96-well plates or sample cups that are placed in the video. We can easily switch between the two and move through 96-well plates very quickly. In addition, we only need 100 microliters of sample, so this works very well for small-scale processes where we're analyzing a lot of conditions across very small sample sizes. Lastly, there's also an open platform connection, and so this more importantly allows for instant data analysis so that we don't have to wait around for the machine to finish or insert a USB or other means in order to get the data very quickly. All of this enables our team to move very rapidly. So again, I just want to highlight the open platform connection or OPC that allows the data to move very instantly. So the connection to our internal servers allows us to immediately process data as it uploads. So of course we start with the FAST CDV and data is uploaded as it's generated. So through the OPC, the data is transferred to our internal servers where we're then able to access this data and immediately start processing it and look in real time at, as the trends are coming in, at what the viable cell densities are, how the growth is performing, calculate doubling times, and look at several other metrics that allows us to determine which of our factors from our DOE is the most influential. So in this example experiment, we are looking at optimizing vitamin concentrations in a CHO-based media. So using JUMP, we created a fractional factorial looking at 3 vitamins and multiple concentrations of these vitamins. We utilize the NIST-CHO monoclonal cell antibody producing cell line that's available from the NIST. And the cells were grown in tube spin bioreactors and sampled daily for growth on the FAST CDV. In this slide, what I'm highlighting here is how we use JUMP scripts to automate our data processing. So we start with a script, which is shown here. It won't run, but this example is to highlight how we utilize our tools to automate this process. So using a script for JUMP, although there's other statistical software that will also work with us, such as R or Python. So using any sort of rapid automatic processing tool, we're able to collect the data directly from the machine as it's generated and then immediately plot this. So in this example, we have all of our different formulations looking at the different vitamin concentrations. and how the cells are responding to these different vitamin concentrations in a batch experiment. So we're able to plot this data across the process time that's shown on the x-axis and look at the viable cell density from the FAST CDV and see how the trends are changing over time, and then determine immediately which is the most important factor from our design of experiment. So in addition to the viable cell density that we're able to track over time, we're also able to analyze the viability or the viable cell diameter that are also captured by the FAST CDV and plot these over time. So again, using jump scripts, we're able to directly pull this data from the machine and track the viability and see any deviations perhaps that might occur along the way, and track how the viable cell diameter is also changing over time, which will ultimately tell us how our cultures are performing and how each of our factors that we're analyzing from our design of experiment are influencing these things over time. So all of this data is automatically generated by the FAST CDV, and we're able to immediately pull it with minimal effort. So to sum up this first part of the talk, what I've shown you is how the FAST CDV enables rapid data processing for media development. What we love is that we only have to use 100 microliters, so low volumes are required, which enables rapid screening of small platforms and high throughput. And again, the high throughput from the 96 well plates, so we're able to automate all of these cell density measurements. All we have to do is perform the sampling, and then we are able to rapidly analyze our data from the network integration. So the OPC allows for rapid data analysis as it uploads to the servers and allows us to directly pull that data into our own internal analyses. And then the machine also operates with high accuracy, so high resolution images and enhanced counting for accurate statistical analysis, so we can really rely on the data for the most robust data analysis and see how our trends are performing over time. Okay, moving on. So now we're going to move on to our second use case. So looking at accelerating analysis of multiple cell types. And why this is important to us is that with media development, for Lucius Labs, we work with a lot of different cell types. So whether these are for fibroblasts or epithelial cells, these cells all behave very differently in culture and also when they're being counted. So to highlight here, so of course, we look at multiple different species. So this comes with its own challenges, where all of these cell types come in different shapes and sizes. And more importantly is that for your data collection, that this requires tuning and inspection of the cell types. And ideally, you wouldn't be sacrificing culture volumes in order to do this. So highlighting here, suspension cells, of course, have very nice spherical morphology. Adherent cells, when they're spindle and elongated, tend to take on a different shape when they're being imaged. And then, as well as clump cells. These, of course, are very hard to accurately count because it's hard to see how many cells there exactly are in the clump. So how do we enable accurately counting each of these different types? So with the FAST CDV, there is the ability to create cell inspection profiles that are tailored to each cell type. The software supports several inspection cell types, and what is highlighted here are the parameters that can be adjusted in order to accurately measure viable cell density, viability, and cell diameter. By adjusting parameters such as the live cell minimum size, we can change this for each unique cell, as each unique cell has its own, or each unique cell type has its own minimum size. We can also adjust things such as the intensity of the dead cells, the intensity of the live cells, and all of these are critical parameters in order to capture the most accurate information for each process, each cell type, and of course, each media condition that we're running. So we're able to do this for whatever cell type that we're choosing to measure, and then we can, of course, change these if they are needed to using what is, I think, very unique to this instrument, or the reanalyzer function. So the BioProfile FAST CDV reanalyzer function enables rapidly tuning cell inspection types without sacrificing cells. So changes to cell inspection parameters can be reanalyzed in samples that have already been recorded by the machine. So what's highlighted here is just a snapshot of what the reanalyzer function looks like. And by just highlighting or clicking on the sample that we choose to reanalyze, we can select a different cell inspection profile and then reanalyze the images that have already been captured by the machine and select an optimized parameters that enable the most accurate information. So the resulting images the resulting changes made can also be directly viewed on the cell images, and all of this information is stored, so you don't lose the initial information that was already captured. You just keep reanalyzing, creating essentially new images that contain new information, but all of the original data is still there. So by using this, we don't have to sacrifice our precious culture samples. We can simply just reanalyze everything and create the most optimized cell inspection type. So in this example, what I'm showing is how we ultimately tuned our cell inspection using the reanalyzer function in order to increase the number of dead cells counted, or essentially reduce the viability. So we did notice that there was a few cells within our cell type a few dead cells within our cell type that were not being counted. So the viability was higher than what should have been reported on the machine. So by changing some of the parameters within the cell inspection type, we were able to refine this and ultimately land on the most accurate viability that we could determine, and the most accurate average diameter. So what you can see here is that by multiple iterations, we refined the viability, so going from 99% viability down to 90.4% viability. But what really didn't change was actually the viable cell density. So we weren't actually changing the number of viable cells that were being counted, but what we did change was the number of dead cells that were being counted. So we were able to refine this and increase the number of dead cells that were accurately being determined as dead cells. And so this reduced our viability, but we were a lot more confident that we were capturing the most accurate viability, but weren't actually changing what the viable cell density was, what the average diameter was. We were simply just capturing more of the most accurate information. And all of this was done by just using the reanalyzer function. We didn't have to sacrifice additional cell culture volume. However, we did verify that this cell type that we landed on was the most accurate across our samples. So to summarize the second part of the talk, what we've shown here is that the FAST CDV accurately measures a wide range of cell types for adherent and suspension workflows. So really any cell type can be taken and accurately counted on the machine, as there is a high resolution images are captured, and this is able to detect all of the unique aspects of all cells. And using some of the advanced algorithms that the machine uses to accurately count each image, we're able to accurately determine what the viable cell densities are, what the viability is, what the viable cell diameters are, and adjust these based on each cell type, each unique workflow, and make sure that we are counting or accurately counting what the viable cell densities are. And a part of this workflow is, of course, the reanalyzer function. So this enables us to change each cell inspection profile and determine that we are accurately counting what we should be, or not counting what we should be. And this function is able to be used without sacrificing additional culture sample. so we can accurately determine what should be counted, what shouldn't be counted, and we don't have to keep running through cell culture sample in order to determine this. So all of this is really critical to rapidly determining how we are able to count cells without having to do a lot of work around optimizing different workflows. All right, moving on to the last part of the talk, we'll focus now on the accuracy of the machine across cell densities and how we are able to use it in fed batch modalities. So working across a lot of cell densities, this enables, of course, different challenges, as very high cell densities can sometimes be challenging to accurately measure. And so in our case, we work a lot with fed batch processes, which of course can reach very high cell densities. And this requires accurately measuring at these, these high cell densities. So what's shown here is just the schematic of, fed batch over time versus batch. So the viable cell density over process time in fed batch is usually able to reach much higher viable cell densities and for longer periods of time, as what is supplied during fed batch is, of course, feed. And so we work a lot to develop feeds and determine what is going to be the most optimized feed and at which point feeds should be supplied to the culture. So the FAST CDV enables us to determine along our processes when we should be feeding, how we should be feeding, and how the cells are responding to the feeds, of course, is one other critical parameter to this. So for this example, this was a fed batch experiment to qualify feed response, and we used the FAST CDV across all cell densities. The key challenge here was to address whether we were able to accurately measure viable cell density over a wide range of cell densities. So this was performed in suspension fed batch using a CHO cell line with a feed that was supplied at 2% every day over a course of 9 days, with a viable cell density ranging from 0.5 to 20 million cells per mil. This is an example of our control feed response of where so what's shown here is a growth curve of the viable cell density over process time. So showing just a typical feed response that we were able to plot with data from the FAST CDV. However, our key question here was whether or not the FAST CDV was accurately counting cells across each cell density. So what we did was to look at the viable cell density that was measured by the FAST CDV and plot this to a reference instrument viable cell density. What is important to highlight here is that no dilution was required at densities over 10 million cells for the FAST CDV. However, our reference instrument does require manual dilution for densities over 10 million cells per mil. However, what we saw was that between both instruments, there was a very tight correlation in viable cell densities. So this ultimately told us that the FAST CDV is accurately measuring viable cell density across every cell type, and that this correlates very well with a reference instrument. So we can reliably determine that what we are counting is accurate as determined by another instrument. Furthermore, we felt very confident that we can accurately measure viable cell density in fed batch experiments. So utilizing the FAST CDV is a very nice option for us, as we can use a high throughput platform for something that doesn't require a manual dilution over 10 million cells per mil. So in the last little bit about the FAST CDV, one of the key highlights for this instrument is, of course, the inline dilution. So there is, of course, with some cell densities that there may be the requirement for external dilution versus an internal dilution. With the FAST CDV, there is an inline dilution if there is the need for a dilution. And of course, manual dilution from external source can introduce additional prep time as well as variability from introducing pipetting errors. With the inline dilution of the FAST CDV, there is the elimination of the time that it would take to prep this, as well as introducing pipetting errors from manually doing this yourself. So in this example of the workflow, the concentrated cell sample is internally diluted using the FAST CDV's own solution, and then this is mixed and then analyzed by the machine. So this rapidly moves from just sample to process sample without the need for manual dilution. So when determining whether the inline dilution maintains accuracy, what we did was look at the viable cell density across 3 dilution factors. So the same sample was used to compare dilution of no dilution to the inline dilution, and then the linear regression of viable cell density and dilution factor was plotted. What we saw was that there was a poor R-squared value, suggesting that there was no correlation and no response from the viable cell density with the change in dilution. This also suggested that the dilution was not necessary, at least for the densities that we tested. So we can accurately rely on both the dilution factor and no dilution when for the some of the densities that we tested. So this is really nice, as it does speed up a lot of the processes where we don't have to consider if the cells need to be diluted, but even if we did, there would be really accurate measurements using the inline dilution option from the FAST CDV. Okay, so to sum up the talk today, what we've shown you is that how our team utilizes multiple tools to increase the speed at which we develop cell culture media, and how the BioProfile FAST CDV is an important tool for our team. For media development, our team is creating several different media formulations in a wide variety of cell types, and it requires a high throughput option to swiftly move through experimental design to data analysis. So the FAST CDV is an important part of this, as we're able to quickly move from our DOE and data collection and immediately plot the data by using the OPC integration. So there's also a high throughput capability by using 96 well plates, and low volumes are required, which is essential for high throughput and small processes. And of course, the network integration works very well with our JUMP software, so that we're able to immediately determine which is the most important factors for our media development. We've also been able to test across multiple cell types and create several different cell and spectrum profiles, no matter what cell type we're working on. So we're screening media for adherent and suspension cell lines, and thus we need accurate cell counts for each one of these. What's really nice is that without sacrificing samples or cells, we're able to create cell inspection profiles for each cell type that we use. Of course, we're able to use the reanalyze function as a key part of that, which doesn't require sacrificing cells, and then we can move on to just generating data. And then lastly, we've demonstrated that there is a high range of accuracy of the machine. So looking at a wide variety of cell densities, as well as inline dilution versus manual dilution, that the machine has very high accuracy and is able to accurately determine what the viabilities are, and what the viable cell density is. What's really nice is that there is the inline dilution, so whether cells need to be diluted or not, there is no need for external dilution. And then within at least what we've determined from 0.22 to 25 million cells per mil, there was no dilution required, so there was very high accuracy within these cell ranges. And all of this enables our team to move very quickly and develop cell culture media solutions at a very rapid pace. So thank you everyone for tuning in. So we're very happy to talk with anyone who's interested after this. Please visit luciuslabs.com or follow this QR code if you would like more information or to determine how we can provide more solutions for your team. So with that, we will take any questions.
Well, thank you very much, Sophia and Elizabeth, for that great presentation. I hope everyone enjoyed it. And here's Elizabeth and Sophia again to join us now for the Q&A portion of the webinar. So remember, if you are watching this via LinkedIn Live events, please go ahead and send your comments through the comment section. And if you're watching this via GoToWebinar, please go ahead and send those questions in that questions window there. Now, while you were speaking, I did receive a bunch of questions, so let's start with this first one here. And it looks like they're addressing this to you, Elizabeth. This audience member is asking, what are the biggest bottlenecks you encounter when collecting viable cell density data during development, and how can automation help address them? Great question. What are your thoughts on that one?
Indeed, is a great question. So the most common bottlenecks are manual sample dilution, inconsistent counting methods, limited throughput, time required to transfer and analyze the data. And automation helps by standardizing workflows, eliminating manual dilution and data entry, delivering faster, reproducible results. So this allows the researcher to make decisions that accelerate cell culture development and process optimization.
Thank you very much for that. Let's go to the next question here. This audience member is asking, can you share an example where rapid viable cell density measurements enabled a larger or more efficient design of experiment study than would have been practical with traditional methods? Who would like to take this question? Is this a Sophia question?
Yeah, I can take that one.
Okay, thank you.
Yeah, I think there's a lot of, so going back to DOE, there's a lot of screens that we do. So these are usually called definitive screening designs. Well, we're looking at more than 10 variables, which can easily create over 60 conditions for us. And if we run these in triplicate, we're well into over 120 different samples that we have to run. So using something like a 96-well plate is really pivotal for us, as we can move through an entire plate without having to go back to the machine and add more samples to it. So I think that eliminates kind of some more traditional methods of where, for like, if you think of a hemocytometer slide where you have to keep adding new samples to it, we don't have to do that. We can just kind of load the plate and then walk away.
Perfect, thank you. Don't go anywhere. I think this next question is for you too. This audience member is asking, the FAST CDV offers inline dilution capabilities. How does that feature help researchers accurately measure cultures that span a wide range of cell densities during the media optimization workflows?
Yeah, I think kind of what was highlighted in the part of the presentation of where we're looking at fed batch experiments, those can start at low densities and then go all the way to very high densities. So that enables, I think, really, so the inline dilution enables really looking at any type of experiment, whether you're working with low densities or high densities. That inline dilution option can flex between having looking at something where there's low densities to looking at something that's high densities without having to change too much on the instrument.
Okay, thank you, Sophia. Let's go to the next question here. This person is asking, beyond faster cell counting, what impact can automated cell analysis have on downstream data analysis, decision-making, and overall media development and manufacturing timelines? Who would like to take that question?
I can answer that. So the automated cell analysis transforms the entire data to decision workflow quicker, and this impacts faster data availability to make that decision, reducing manual data handling that minimizes the transcription errors that can happen, digital connectivity and automation together to streamline data collection, visualization, and analysis. Higher throughput is another factor that automation helps. And then earlier detection of process deviations, all of which contribute to enhancing that timeline for process development.
Thank you very much for that, Elizabeth. Okay, this looks like it's our last question. This audience member is asking, what would be your advice for a researcher who wants to pioneer cell culture work and experiments in a low and middle income country? I'd like to take that one. Is that a Sophia question?
Yeah, I can definitely start.
Okay.
So I think, my advice would be to utilize a lot of the information that's already freely available. So there are a lot of media formulations that are publicly available, like DMEM, DMF12. You can go find that recipe and understand each of the different components that goes into it. So there's a lot of just free information from that standpoint. And then kind of getting more into the actual experimentation. I think even just starting with like a small microscope and looking at cells under a slide, using just like iodine. I mean, I think that was one of like the first experiments that I ever did. And it's a great way to just kind of easily visualize cells without having to use a lot of expensive reagents.
Elizabeth, anything else you'd like to add before we close out the Q&A?
No, thank you to everyone for joining us. And if you have interest in this product, the email is there in the webinar chat. Please do write to us.
Okay, perfect. Okay, then I will close out this Q&A portion of the webinar. So thank you very much, Elizabeth and Sophia, for answering all our questions. If we could not attend to your question, rest assured the team will follow up with you after this presentation. And just like Elizabeth said, the email address is right here on your screen, so please go ahead and utilize it if you have any further questions or inquiries, okay? Thank you, everyone, for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive of this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve our webinars. Additionally, I have shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues once they register for the recording here as well. And for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event to gain access to such features as viewing the recording and more. Now, please join us in thanking our speakers for their time here today. So thank you very much, Sophia and Elizabeth. It was wonderful having you on here and for sharing your knowledge. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. Until we meet again, I'm Sonia Hunt. Please take care and bye for now. Bye, everyone. Bye, Elizabeth. Bye, Sophia.
Hey, thank you.
Bye.
Bye-bye.